Count upside down. Press Alt A. What?
Okay, incorporated. All right, so as I was saying, we're going to look at the biological processes that are really the foundation of the machinery for our behaviors and what we do. And so we need to start with specifically talking about the neuron, okay? And the neuron is, if you want to think about it, the most basic unit of information. It's held in the brain, okay? We have over 100 billion just in the brain itself. And I want you to think about this. So it's 3 to 4 gram thing setting in your skull, has over 1 billion cells in it. And its relative size, it actually consumes about 20% of our energy. Okay, so you think about that, that little area right there consumes about 20% of the energy through nutrition, mainly sugar, that we intake each day. All right, that's a significant amount when we think about it. Humans do not have the largest brain. The larger the species is, the larger the brain. And in some, there's actually sub-brains throughout the body. For example, with jellyfish, they actually have brains that kind of wrap down to the tentacles. Squid actually have several brains that control each of the arms, almost a separate brain for each arm. And then as we get into larger mammals, such as elephants, they have a sub-brain near the back that kind of helps coordinate the legs with the front, okay? But if there is a mammal with a large brain, it's elephants because of their size, their pure size. What humans do have that is abnormal among species is we have an actual oversized brain for our body mass. Okay, so most animals in the animal kingdoms, their brain size is in proportion to their body size. Okay, humans are one of the very few species that we actually have extra room that isn't being committed to bodily functions and survival. And a lot have argued that that's kind of why humans have developed this idea of consciousness and all of these kinds of things, because the brain is slightly larger to the body proportions in humans, okay? There's really 2 types of cells that are operating in the nervous system. The first one is, of course, the nerve cell. And it is one of the very few cells that you have that is there from birth to death. Okay, you can grow new nerve cells, but once that nerve cell is dead, it's gone. It's no longer exists, and the likelihood, basically, your age of new ones growing in its place are very, very slim. Okay, so it is one of the very few cells in our body that actually stays the entire lifespan. Believe it or not, with the rest of our body, we get a whole new body if everything was perfectly in sync, and it's not. So you got to imagine this being perfectly in sync. We actually get new bodies every 5 to 7 years because that's how the skin cells die, intestinal and organ cells will die and reborn, bones do the same thing. And they're all on about a 5 to 7 year cycle. They cycle differently with each other. For example, skin actually is regenerating almost daily in certain parts of your body. And that's why, you know, if we if we were to actually look at the air quality in this air in this room or wherever you are, a large portion of it is actually skin cells that we're breathing in from moment to moment. Okay. So, but nerve cells are the ones that they're there until you die. They don't regenerate, they don't do anything. And that means that they need helper cells. Okay. And the helper cells are these glial cells. And what these glial cells do is they clean the neuron, okay, and keep it kind of nutritious and healthy, and kind of fix it every time it starts to kind of break down. Okay. And these are getting increasing amount of attention within the field of neuroscience, because we're finding that there's an association between a person's learning ability and the amount of glial cells that they're producing. Okay. The more glial cells, the more cognitively active the person is even into late adulthood. And these glial cells kind of react to how much you're using your brain system. So that whole idea of lifelong learner kind of idea that you should exercise the brain as much as you exercise the body. It's showing that the more and more mental activity you are, the more glial cells are produced, which keeps the nerve cells all healthy and ready to cognitively task. Okay. And indeed, there's a lot of other studies dealing with individuals with dementia that is showing that a lot of dementias have reduced amount of glial cells in their nervous system. So so they kind of work symbiotically together, and dependent on how many there are and whatnot is what determines the health of the brain. Okay. These neurons, of course, are interconnected. They give rise to intelligence and consciousness, or at least our experience of it. Okay. And so I want you to think about this. We have 100 billion cells, individual cells in the brain, but the number of connections that a cell might have to another one is exponentially that. Okay. How can I put this in literal terms? All right. Despite all the technologies that we have, the supercomputer, I think it's in Japan, all of these modern technologies, okay, it's still expected that it won't be until around 2028 that we'll have a computer that has the processing ability of one human brain. Okay. In fact, right at this moment, the best we could do is if we took everyone's cell phones, okay, stacked them in the state of Montana, filled the state of Montana 2 stories high, all those cell phones would not have the capacity of that 3 to 5 grams that is sitting on the top of your head. And when you think about that, that's pretty incredible. But a lot of people are like, well, why are you not as smart as my cell phone? You got to remember, your brain is controlling a lot more than just your thoughts. It's controlling your heart. It's controlling your lungs. It's controlling how you're sitting in space. A lot of people don't realize this, but when you sit for a long period of time, you're not actually sitting. We move every 5 to 7 seconds, but we don't recognize it because the muscle twitches are so small. But our brain has to constantly control that. Otherwise, like when you start losing that muscle control in older life and you get bedridden, that's why a lot of elderly end up with bed sores, because the brain is no longer shifting their muscular system in order to compensate for what's being crushed, is basically the cells which creates those sores. Okay, so the brain has a much more complicated system to run than our basic cell phones, is what I guess I'm trying to say. And we'll see even more of that, the complexity, when we get to sensations and perceptions in the next section. Okay. But I just wanted to provide some of the complexities of our nervous system. And like I said, despite technology, we still don't have a computer that will process as much as one single brain does. So it's just kind of cool. All right. This will play. Let's see if I can just play it. This volume wasn't working.
to ask questions, answer questions, and post some interesting things, including your artworks. And you can also change the quality settings to the highest one for better graphics. In this neurology video, we will look at Armando Hasurigan, biology and medicine videos. Please make sure to subscribe, join the forum and group. For the latest videos, please visit Facebook, Armando Hasurigan. Please like, and here you can also ask questions, answer questions, and post some interesting things, including your artworks. And you can also change the quality settings to the highest one for better graphics. In this neurology video, we will look at the neuron. The neurons are the communication cells. They receive signals. What you see, smell, hear is thanks to a group of sensory neurons. Neurons also send out signals or information. When we move our hands every time we breathe, it's because of commands being sent via efferent neurons. So neurons are a big deal, and because of this, we have billions of neurons in our body. Here's a typical structure of a neuron. It consists of dendrites, which receive an information, a signal. The cell body, the soma, the axon hillock, the axon, where information or the signal in a form of an impulse is propagated through. There can be myelin, which wraps around the axon to help in insulation and speeding up the impulse. And all the impulse will end at the synaptic terminal, where the impulse, the information, is passed on to a target cell. So looking at it, an input signal is received by the dendrites, gets passed on to the cell body for integration, and then the output signal is released from the synaptic terminal to a target cell for a specific response, a desired effect response. The target cell in this case is another neuron, but this neuron is different in that it is an unmyelinated neuron. It has no myelin sheath wrapping around it. This means that the propagation of the impulse along the axon is much slower. When you have a neuron with myelin sheaths, the impulse travels much faster. But now you might be asking yourself, how is the information from one cell, from a neuron, is passed on to another cell? Well, let's zoom into this area here, where these two cells are close to each other, where they synapse with each other. Here we have part of the synaptic terminal of the first neuron and its presynaptic membrane. And here is part of the dendrite of the second neuron and its postsynaptic membrane. The gap between the first and second neuron is known as a synaptic cleft. In the ends of the synaptic terminal region, like here, we find many mitochondria and vesicles containing what's called neurotransmitters. These neurotransmitters are released for communication, for the communication process between cells. What happens is that when a signal arrives at the dendrites of the neuron, it will create an impulse that will carry this information and propagate it towards the terminal. This impulse is an action potential. The action potential will cause, once it arrives at the synaptic terminal, it will cause the vesicles here to release the neurotransmitters into the synaptic cleft, where the neurotransmitters will then bind onto the cell's postsynaptic membrane. So we can say that the synapse is the site for intracellular communication. And seeing that the postsynaptic membrane of the postsynaptic cell belongs to the dendrite of a neuron, it will receive this information and then create another action potential that will propagate along the axon towards the synaptic terminal. So a new input signal is received by the dendrites. It will be integrated in the soma, and then the information, the action potential, the information will pass along the axon towards the terminal, and will be passed on as an output signal via neurotransmitters that will target a particular cell. It can be a neuron again, or it can be a muscle cell or an endocrine cell, any kind of cell, depending on where the neuron is located and what its desired response wants to be, what effect it wants to cause. So now that we have an idea of how signals are being passed all around our body and how we receive signals all around our body, let's learn more about the soma of a neuron and how the neurotransmitters are packaged up, are made. So here we have a close-up of the soma of the neuron. We have the nucleus containing the genetic material, the rough endoplasmic reticulum around it with bound ribosomes and free ribosomes for protein synthesis. We have the Golgi apparatus for packaging, and we have the lysosome. Now, the protein neurotransmitters are synthesized in the rough endoplasmic reticulum by ribosomes and then packaged up by the Golgi apparatus. So here we have the rough endoplasmic reticulum synthesizing new neurotransmitters that passes them onto the Golgi that will then package them up in vesicles. These vesicles containing neurotransmitters from the Golgi are then brought to the synaptic terminal. Here we have the synaptic bulb of the synaptic terminal that we're zooming into. So these vesicles containing the neurotransmitters and also mitochondria, they move down via microfilaments or microtubules, and they move to the terminal bulb here. The vesicles are in the synaptic bulb where they can be released via exocytosis to the synaptic cleft when an action potential arrives. The neurotransmitters can be reabsorbed from the synaptic cleft and form vesicles, and then these vesicles can be recycled. They can travel back to the soma of the neuron where they were fused with lysosomes. The lysosomes will digest these vesicles for recycling. So neurons are a big deal, what we just talked about, for cellular communication, for sending out signals, and also receiving signals, receiving information. These neurons I just drew is actually a typical structure or typical shape of a specific type of neuron, an efferent somatic neuron, that is. However, there are a few types of structures neurons can be categorized into. And these structural categories, it can be determined by which part of the nervous system the neurons belong to, if that made any sense. I'll just draw a diagram to explain this. So the nervous system, remember, it can be divided into two major parts. That is the central nervous system and the peripheral nervous system. Now I drew two peripheral nervous systems because the peripheral nervous system consists of a sensory division and a motor division, or better yet, an efferent division. The sensory division of the peripheral nervous system consists of sensory neurons that look something like this. It has dendritic branches here, the axon on either side of the soma, the cell body, and then the synaptic terminal here. This type of neuron is categorized under the structure of a unipolar neuron. Now we also have another type of sensory neuron which is slightly similar in that it also consists of dendritic branches, but the dendrite will extend to the soma. Then we have the axon and finally the synaptic terminal. This type of neuron is categorized under the structure of a bipolar neuron. As you can see, the unipolar and bipolar neuron are only slightly different. The bipolar neuron, we have two separate processes separated by the cell body. So we have the dendrite and then the axon on one end. The central nervous system consists of many interneurons. Interneurons, you can say, are neurons that bring signals within the central nervous system, so from the brain to the spinal cord, for example. But we also have interneurons that act as glial cells, helper cells. Anyway, these interneurons have some completely different structures to other neurons. An example of an interneuron is this, what I'm drawing. And it doesn't look like it has an axon, but only has a cell body in the center with many dendritic branches around it. This type of neuron is categorized under the structure of an anaxonic neuron. Then we have a multipolar neuron type, which contains dendritic branches on one end and then straight away an axon terminal branch on the other, separated by the cell body. Some multipolar neurons in the central nervous system look slightly different than this, and we shall soon see what I am talking about. Now, the efferent division of the peripheral nervous system contains efferent neurons, motor neurons, which are all multipolar in structure. And this is the type of neuron I drew in the beginning of this video. We can have efferent neurons with myelin wrapping around it, or we can have efferent neurons without myelin wrapping around it. Either way, both are multipolar in structure, in that it consists of dendritic branches, and then we have the cell body, and then the axon and terminal branches. As you can see, this is the other type of multipolar neuron I was talking about in the central nervous system. The terminal branch doesn't have to be really close to the soma body. In the efferent division, the terminal branch is extended with the axon. Hope that made sense. And anyway, that was it for the neuron video for neurology. I hope you enjoyed it, and hopefully I'll make a video on an action potentials soon. Thank you.
All right, so that was just kind of an introduction to the different cells we have within our system. And there's some areas, and just to kind of repeat, and again, this is on in Canvas, the dendrites are the receiving areas. The soma is kind of like the body, it's a little brain. The axon is where the electrical potential goes down in order to release neurotransmitters at the terminal, the axon terminal, okay? And then again, as I said, not all cells are myelinated, and we'll get to this in a minute, but it's kind of like these little fatty nodes that are on some neurons, especially in the peripheral system. All right, so again, here we have the basic things, and I want to discuss just a few. Let me look at something really quick. Okay, yeah, we are good. Okay, I want to focus on the idea of these terminal buttons and the dendrites, okay? Because they are constantly connecting and reconnecting every time we sleep. That's kind of how our brain remembers things, by making new connections with nerves or cutting off connections with other ones, okay? I want to talk a little bit, because as we get into different sections of the class, these things will need to be brought up again. And we'll start with this myelin sheath, okay? As was said in the video, what this does is it not only just protects the cell, but it also speeds up the transmission of the electrical impulse. So, you know, if we have an example here, let's show how this kind of speeds it up. Is, let's say this little red line represents an unmyelinated sheath, an unmyelinated axon, okay? And again, the electrical pulse starts at this end and goes to the terminal buttons. And so if an electrical impulse occurs with an unmyelinated axon, electrical impulse has to travel all the way down, and you can see in this exaggeration the amount of time it takes. But when you have a myelinated sheath, a myelinated cell, it doesn't have to go down, it actually jumps. So we want to look at this, how much quicker this is to the thing. You can see that it's much quicker than the unmyelinated one, okay? So what areas does this become important? Okay, well, we know from recent studies going back to, I believe, around 2010, we have actually figured out that this occurs in adolescence. The majority of our brain becomes myelinated within about a 4 to 6 month period as we go through puberty, okay? Which means that in childhood, we have a much slower brain And then if you want to think about this, we have a brain that's going about 25 miles per hour. And then within a 4 to 6 month period, we all of a sudden have a brain that's going 2,500 miles per hour. Okay. And so this rapid increase in brain speed, we have started to kind of question, well, what does that mean? And the more and more evidence we're finding is adolescents who have difficulties, we usually say they're hormonal because of all the rush of hormones going through the body. We're finding that difficult years for some adolescents isn't because of hormones, it's because of the brain speeding up, okay? And when talking to most adolescents, when you said, why do you do that? And they say, I don't know, they probably actually don't know, because their brain is going this fast and they're trying to catch up with it as this brain system becomes much, much more rapid and fast. And we find an interesting connection with this, so we'll talk more about this when we get to the developmental section, is what's the difference between adolescents that do really good in adolescence and get along fine, and those who have a really hard time with it. Well, a study on parenting actually shows that there's a difference in parenting between adolescents who tend to do good, those who tend to do bad. Again, there's always variability in this, so it's not 100% proof. But what we found is adolescents who had a difficult adolescence had a type of parents who said that the adolescent gets in trouble. The adolescent says, I don't know why I did that. And he says, well, if you go figure it out, your Xbox is gone until you can come tell me why you did what you did. Okay. We find that an adolescent that goes through that kind of parenting tended to have a more difficult adolescence. Okay. We found that with parents who, when the teenager says, I don't know, or the adolescent says, I don't know, they had a parent who sat down with them and said, well, let's go through it together. And they start from the beginning of the situation and work through what happened. We find that those adolescents tended to have an easier, quote unquote, adolescence without much of the chaos. And what is the parent doing from a biological perspective? They're trying to get the adolescents to understand what the brain system is doing in that much faster area, okay? And then the second question is why in early childhood do we have a less myelinated brain, but then when we go through adolescence, we have a much faster brain? And we believe this is because in order to learn really good, you have to have a slower brain. Okay, so remember what I said about kids between the age of 0 to 6. On average, they'll learn around 50 to 70,000 words. Okay, think about what it would take, a system that it would take in order to learn all that. And it's needing a much slower system where connections can be made much tighter and closer together. Okay, however, if you think about this from an evolutionary perspective, once you go through adolescence, you get your physical adult body. And let's say there's a 5-year-old in the street and there's a car coming towards that 5-year-old at 50 miles per hour. Okay, the 5-year-old is probably trying to make a decision whether or not they should move or not. The caregiver instantly reacts and goes and gets the child out of the way of the car without thought really processing it. And that's the advantage of having a faster brain. And it kept us, if you want to think about ancient times, it kept us from being eaten by tigers and wolves and all those kinds of things. And now and it helps us protect our children in instant reaction moments. Okay? So that's kind of why we're thinking this myelination occurs later on after the solid learning years have taken place. And then another aspect of the myelin sheath that is kind of important and we'll talk about is dealing with a certain type of dementia called Alzheimer's. Okay, You probably have heard that it's one of the most rapidly growing type of dementias. I should state that it is a subtype of a dementia. When we get to the section on memory and stuff, we'll see that there's probably about 15 different types of dementia, and Alzheimer's is just one of them. Okay. But what's happening, we found with Alzheimer's, is around this myelin sheath, a black kind of tar starts to envelop it. Okay. And basically what it's doing is it's suffocating that cell. Okay. Because a cell, like everything, has to release sodiums and all of these things. The glial cell has to have access directly to the cell to clean it. But this black tar starts to just wrap around it and suffocate the cell till it dies. Okay. And that's why it's a progressive disorder, because it really only needs to affect less than 1% of the brain cells. Because what happens is when that one dies, and let's say another one dies that's close by, it will start to kill the cells because they're no longer receiving signals from those two cells. So the other cells don't have Can everyone still hear us? Online?
I can't hear Mr. Peterson. I can't hear the teacher.
Switch to this. I hope so. Let me get this slightly. Anyways, as I was saying, while this is rebooting, is it basically suffocates the cell, and then the cells that can't communicate with anymore also die because they're not receiving any new messages. So they figure, don't need to be here anymore. Okay. And that is when we get to certain types of dementias, and Alzheimer's is an example, is it really is a disease that just suffocates the cells to death, is basically what they are. No. Okay, while we're bringing this up for you guys. Sorry, everyone online. We kind of had a power outage that kind of shut us down here. So we're just getting booted up again. All right. Sorry about that. All right, here we go. So the thing that I want to talk about next is the neurotransmitters, that the communication. So our nervous system is what we call an electrical chemical event. Okay. The chemical part is when a neurotransmitter is released into the synaptic gap, and then it binds with the nerve that is next to it. That's the chemical part. If there's enough medication, enough neurotransmitters received, then that next cell will fire. Okay. And again, we'll talk about this, especially when we get to the abnormal psychology section. Okay, because a lot of the antidepressants and anti-anxiety medications that are out there are meant to increase the amount of certain neurotransmitters. Usually with depression, we use what are called SSRIs, which is serotonin, something reuptake, blah, blah, blah. Basically, what it does is it blocks it so that, for example, serotonin cannot be reabsorbed into the ascending cell. And what that does is it then ends up putting more serotonin in the synaptic cleft and increase the amount of serotonin a person has available to it, instead of it being reabsorbed back into the cell like was described in that video. So we'll talk about that when we get to places that deal with certain kind of medications and everything. But I want to talk about some of these neurotransmitters, because each one has a specific purpose. So for example, acetylcholine participates in mostly movement and automatic functioning and learning. So those automatic processes that are going to occur in the limbic system and the spinal cord, and the movement of those deals with acetylcholine. So a lot of runners and athletes actually will take supplemental acetylcholine because it helps promote the muscle growth and the movement that the athlete is doing. Okay. Dopamine will be of really particular interest in two areas of this course. One dealing with motivation, because dopamine is the reward chemical that's in the brain. And we'll also talk about it when we get to certain disorders. Okay, what do I mean by dopamine is the reward? If we look at drugs that target dopamine reception, we're talking about things like cocaine and methamphetamine. Okay, those are what work on the dopamine system. And so if I were to put it into terms of, and I think I've actually mentioned this in the class, the time that we most naturally produce dopamine, okay, is either when we're having an orgasm or we're hitting someone across the face. Okay, those two events actually are the highest levels of dopamine that you can naturally produce. Okay. Now, if you want to know from at least a chemical perspective why things like methamphetamine are so addictive, okay, is methamphetamine quadruples that amount that you produce while having sex or hitting somebody. So it's a very, very intense high. In fact, when you talk to a lot of people on methamphetamine, they can no longer have sex unless they're high, because it's just not a rush anymore. Okay, so that's an example of the importance of this system as far as a motivation system. And like I said, we'll see this come into play when we talk about certain disorders, such as schizophrenia, which is a disorder that deals with disorganized thinking. Usually it's most popularly known for the hallucinations it can produce. Interestingly, the biggest hallucination is actually olfactory. They smell things that are not present. Okay. With that being said, if you smell bacon and bacon is not cooking anywhere, you're not having a schizophrenic moment. You're actually having a seizure and there's a blood clot in your brain. That's one of the most common signs that you're about to go through a seizure, is smelling bacon. Unless it's a chronic disease, like some people's seizure disease that a lot of people have. Just saying, if you smell bacon and it's not there, call 911. But anyways, so schizophrenia, the one thing we know about people with schizophrenia is their brains are basically marinating in dopamine. okay? Especially when they're active during their disorder, they have an excessive amount. And that's why when you hear people who are on meth, they have hallucinations, they have a lot of the symptoms that you see with schizophrenia. Okay? Now, a lot of the drugs that are used for schizophrenia are meant to drop down the dopamine levels, bring them down to a more controllable amount. Okay? But there's another disorder called Parkinson's disease, which is a movement disorder. Okay? Parkinson's disease is associated with really, really low levels of dopamine. Okay? So the medication used for Parkinson's is aimed at increasing the amount of dopamine in the brain. And this becomes the trade-off when you start treating both of these disorders, because the major side effect of schizophrenic medications is Parkinson's symptoms. The major side effect of Parkinson's medication is schizophrenic symptoms. So that's why when people are talking about should we have the family member medicated or not, that's kind of the decisions they're trying to make, is what's worse, Parkinson's or schizophrenia, when it comes to medication treatment. So what else will we talk about? We'll talk about dopamine when we get to romantic relationships, because what we find is there has to be timing with certain chemicals in the body, and dopamine has to be the last one if you're actually going to fall for someone, at least from a chemical perspective. And then GABA, it's a neurotransmitter that exerts inhibitory control over the central nervous system. So if you have a family member with high blood pressure or heart disease, they usually give a GABA inhibitor because it starts to slow down the amount of signals that are going to the heart and help regulate it. Okay, and so it's meant to kind of calm down the system. Glutamate is also another popular one that deals with learning and memory. A lot of medications that are meant for things like memory loss and stuff are actually glutamate based. Norepinephrine, this is an excitatory neurotransmitter. This is what kind of gets you up and gets you going, and really helps you stay aroused and vigilant. Okay. And then serotonin is an interesting one because it's often associated with mood. Okay, the main serotonin area is right around the amygdala, and the amygdala is associated with a lot of mood and feelings and emotions. Okay, and this is why things like with depression and stuff like that, we find that increasing the amount of serotonin often relieves depressive symptoms. Okay, but it does get a lot more complicated than that, as we will see later in the course. Okay, interestingly, when we look at how these play roles in different things, well, let me talk about these really quick. So neuropeptides are kind of work with neurons to help regulate. So, for example, pain and stress and endorphins working off the pituitary gland relieve pain. We actually have one that's not included in here is opioid receptors. Okay, and again, this is another one. We talk about pain relievers that are opiate based, and why they can be so addictive is because they work on this opiate system. And so does things like nicotine with cigarette smoking, because what this is it's a pain relieving system of the body. Okay. All right, so let's talk about for just a minute the brain system itself. Okay, and really we can divide it kind of into two spaces. If we take here This is the limbic system. We'll kind of include the cerebellum for now, and we'll include the brain stem. All right. So why is this important? This is the most basic part of the brain. In fact, some refer to it as the reptilian brain. Okay. Because any species that has a brain has a limbic system and a brain stem, right? So just as being a member of the world that has brains, this is something that we have across species, right? What tends to differ among species is this cap, this cortex that sits over the limbic system, okay? This cortex is what gives rise to our perceptions and ideas, our control over our own behavior. And this is kind of an interesting area because dependent upon the species specialization will determine which areas of the cortex actually are larger, okay? So for example, back here we have the occipital lobe, all right? So birds of prey who need incredible eyesight, their occipital lobe is actually larger than ours is compared to their body size, okay? And different areas of the brain, dependent upon that species specialization, determines what area of the cortex is larger, right? For humans, compared to other species, our area is actually the frontal lobe that tends to be oversized. This is kind of the newer area of the brain. It gives rise to logical thinking, problem solving, social engagement, defining who you are as an individual. When we were talking about psychopaths, this is what's going to inhibit the instant reaction of the limbic system, okay? Interestingly, though, this actually is the last part of the brain to fully develop. The brain develops from the back forward, okay? And then progressively that area becomes fully developed. Based on some research, it's not really conclusive, but it suggests that around the age of 22 to 27 is when this area of the brain becomes finally its fully developed self, okay? What else should we say about this? Well, we'll get to the rest when we get to developmental psychology. This brainstem area really works on all your autonomic processes. So this regulates things like food and hunger. It regulates heart rate, breathing. All of that is within this brainstem area, okay? Probably within the area of social psychology, what fascinates us about this area is research that shows, so what happens when we show you pictures of someone you're really in love with versus just random pictures, okay? And we measure how much you're in love with that person. Well, if you follow the research on love in psychology and even sociology and anthropology, for centuries, love, intimate love specifically, was seen as an emotion. It was an emotional experience, okay? And so what area of the brain that should be lighting up is the amygdala, which is located right here, okay? Because this is the area that is associated with deep emotions, whether positive or negative, okay? What was interesting is that's not the areas that light up. The areas that light up is in this reticular formation and in this paralimbic system that kind of is just in between the cortex and the limbic system. This is a dopaminergic area, meaning that you're being rewarded for thinking about that person, okay? This is a drive section. Okay, the reticular formation specifically deals with hunger and thirst. And so this has made it so we have to rethink what happened theories of intimate love, because we can't call it an emotional experience anymore. It's a drive. It's a basic human drive, just like eating and drinking. Okay. And other studies have shown this as well, that were non-neural. For example, did I mention isolation in World War II here? The isolation studies where the person will actually start to starve to death if they have human contact, those kinds of things. So those behavioral research kind of reinforce this idea that this is a drive, which makes sense why we go so crazy over someone when we have them, in the beginning at least. And then when we lose them, we even go even more crazy, okay? Because if you want to think about it this way, why do we become so obsessive when we lose someone, like get dumped or something like that? It's because basically your food has been taken away, your water has been taken away, and this reticular formation of the brain is going crazy, okay? And if you think about starving, when someone's starving, they'll do just about anything to get food. If someone is dying from thirst, they will do just about anything to go get that quenching water. And now we can explain why people go so extent when they're dumped or when they lose someone, because basically you're starving that area of the brain or the body. So just kind of some interesting stuff dealing with that area and what we've found over the last 10 or so years. Okay. So I also want to talk a little bit about the cerebellum. Okay, some people call this the miniature brain. What this area of the brain really deals with is coordination. So things like language and use of words and those types of things are all coordinated through the cerebellum. Okay, and so some real life consequences of having problems with your cerebellum is the loss of the ability to talk straight, to be able to walk, okay? So if you see some of the kind of, for example, old school boxers, and they have trouble walking, most of them have to use a cane, and they have a real trouble with speech. They can't coordinate their speech well. Well, the reason that is that this cerebellum sits just right at the bottom of the skull, right here, okay? And damage to the brain doesn't happen where the skull was impacted. If you're hit here, the area of the brain that is at most risk for damage is the back area of the brain, because you're basically shoving the brain up against the back of the skull and squishing the cells there, not the ones in the front, okay? And what happens is the inside of the skull actually is not smooth. It actually kind of has roughness, it's sandpaperish almost. And with repeated hits, what we found is that the cerebellum is basically getting sanded off with all those repeated hits to the face and to the head, resulting in uncoordinated movement 10 years later, or ununderstandable speech later in a boxer's career. Okay? So that's an example of how the cerebellum system works and what happens if it has repeated damage to it. Okay? Right. Let's just get through the cortex real quick, and then we will call it a day for today. And finish up with the divisions of the nervous system on Wednesday. Okay. So as I mentioned, the best way to kind of think of the brain is in these divisions. You have your frontal cortex, you have your motor area with this motor strip, and then you have the sensory area, including the somatic area of the brain. And then you have the visual area, which is this back area. And then you have the auditory area. which is right here. Okay. So how do these areas work together and whatnot? Well, this motor strip right here is the areas that is associated with different parts of the body. In fact, we can actually map it on here. And dependent on the amount of nerve communication that has to happen with that part of the body determines how much of this strip is taken up. So, for example, the legs wouldn't be longer in this strip. Maybe the legs would take up this amount of space, okay? While your face actually would take up about this much of the face, almost double what your legs require, because of all the facial muscles and everything that we have to use in order to smile and do all the things that we do, okay? These work together because it sends us sensory information, and then the motor cortex sends to the body what the body should do, okay? And so these two systems work to help the brainstem and everything coordinate our desired movements, okay? This somatic area processes things like depth. It processes things why we're located in time and space. When this area becomes damaged, people tend to have trouble coordinating movement and understanding where they are in space and time, okay? Interestingly, studies of people who are really good at math have an interesting process, because usually when we're doing a math problem, we use this frontal cortex or this logical area of the brain, okay? But people who are good at math and are just kind of those naturals at it don't use very much of their frontal cortex for it. They actually use their somatic lobe, meaning, as it was described to me by people, is they don't see like, you know, how we're supposed to do math. You're supposed to do the first line, the order that you're supposed to do them in and whatnot. They don't see that. They see numbers in their mind interacting with each other. And it's almost like watching a TV show for a person who is really good in math, is because they see the numbers interact with each other. They don't use a logical order to solve the problem. They just see it happening. And when we scan their brains, they're using this spatial area of the brain, not the frontal cortex of the brain, to solve math problems, which is kind of interesting. And we find the same thing with a lot of people who tend to be natural experts in something, is they tend to use that area of the brain rather than the other logic areas of the brain. So something kind of interesting. And then finally, well, almost, is the auditory system. The interesting thing about the auditory system is, if you remember, I said that all sensory information goes to the limbic system first, and then to the other areas of the brain. The auditory system is the only sensory system that is directly connected to the lobes of the brain. So it doesn't necessarily have to go through the limbic system before we actually start processing it with the lobes, okay? Some of the areas that are important and should be noted are the primary auditory cortex and the auditory association areas. Humans have a huge ability for face and name recognition. And it's this area of the brain that actually tells, you know, when you look at someone and you see them and you can associate their name, okay? This is that kind of area that is association. And then this area, this little tiny area, has to do with gustorial responses, meaning choking or disgust, okay? Which we will talk about a lot when we get to emotions and why we're attracted to some people or not other people. It's kind of interesting when you put it, again, if you put people in the brain scan and you put pictures of somebody that somebody really hates. Again, you'd think it was the amygdala that would start the fire, but it's actually this little disgust area the same one that when you taste something nasty, you choke it out, is the same area that is active when you're around someone that really annoys you, which has implications, as we'll see when we get to that section. Okay. Let's stop here and ask, if any